Okay, next up we have Dr. Daniel Hermens, and he will be talking about tracking the associations between environmental factors and structural connectivity changes in adolescence.
Thank you. Good morning, everyone. Thanks for being here, and thanks to the team for the invitation to be involved with this symposium. It's been wonderful so far, and you've all set the scene perfectly for me in terms of adolescent brain development. It's my pleasure to present some findings we have from the Longitudinal Adolescent Brain Study, which has been underway, taking place in the Sunshine Coast region in Queensland. For those of you not familiar with the region, it's about an hour and a half, 90 minutes drive to the north of here. And it is Gubby Gubby land, and given that, I pay my respects to the elders, past, present, and future of the traditional lands that we're on today. In terms of disclosures, I have no disclosures, but I would like to acknowledge or note the funding I currently receive, and that is from the Australian Commonwealth Government and also Queensland Mental Health Commission. I'm going to focus on white matter development in the context of social connectedness, and if there's time at the end, I'll touch on what we've been doing in terms of white matter development and the role of sleep and also early life stress. I'm sure, as many of you know, adolescence is a critical period of social and neural development. Brain regions that process social information are continually developing throughout adolescence as young people learn to navigate their social environments. Social connectedness is a sense of emotional closeness and belonging. It's a crucial environmental factor. Those with increased social connectedness identify more with others. They're in social groups, more supportive and meaningful relationships, whereas those with decreased social connectedness feel more distant, misunderstood, actively avoid or uncomfortable in social situations. We've had a series of papers from labs where we've looked at the relationship between social connectedness and mental health outcomes. We found that decreased social connectedness increases the likelihood significantly of developing severe psychological illness. We've also found that social connectedness moderates the relationship between mental health symptoms and suicidal thoughts and behaviours. And for those who have increased social connectedness, it is protective. And similarly, protective in terms of mental health outcomes for those that experience cyberbullying. So we do know, that social connectedness and white matter development are key features of adolescence. And when I'm talking about the white matter development for these studies, I'm referring to utilisation of diffusion tensor imaging. I'm sure many of you know about that, and those of you who are not familiar with it, it's a technique where we can measure microstructural properties of myelinated axons. It gives us various indexes of fibre size and density or strength of connectivity and degree of myelination. Fractional anisotropy is the most commonly reported measure. It's generally a measure of the direction of diffusion, and seen as being a marker of the degree of axonal packing. Less used but important measures are radial and axial diffusivity, which indicate radial is perpendicular to the axons and tell us about change in myelination or myelin integrity, whereas axial is running along the axons and tells us about axon numbers and coherence. And mean diffusivity is not commonly used in this area, but that's an indicator of tissue density and water content. So there is evidence in the broader literature about a relationship between structural connectivity and social functioning, but there isn't that much literature exploring the neurocorrelates of social connectedness in early adolescence. And so we think there's a need to better understand the role of white matter integrity in this regard in developing adolescent brains. So our assumption is that neurobiological underpinnings of social connectivity likely depend on the efficiency of neural connections throughout the brain. But the big question is what comes first, or whether these things are bidirectional. So social connectedness and maturation of white matter may be bidirectionally associated, and we need longitudinal evidence to determine that. So this is it's a busy slide. I'll walk you through it. This is the protocol for the Longitudinal Adolescent Brain Study, which has been underway at the Thompson Institute in the Sunshine Coast over the last 7 years. We recruit young people from the general community from age 12 and in grade 7. And we do assessments on them every 4 months for up to over 5 years. That is 15 time points in total. And as Tiffany mentioned before, we've got 180 people in the study so far with almost over 1,000 time points and almost 1,000 MR scans. So the assessments, I'll just walk you through actually from right to left. At each time point, we do a structured clinical interview, screening for around 30 disorders. We do a self-report questionnaire. It ranges a whole range of measures from including lifestyle factors and mental health and wellbeing outcomes. On alternate time points, we do different cognitive batteries, so odd and even number time points. We alternate the cognitive battery. We also undertake EEG, resting state and event related. And our MR protocol over 1 hour includes T1 weighted structural scans, spectroscopy, diffusion weighted imaging, and resting state functional MRI. So I'm just going to focus now on 2 studies. One's published and one's about to be submitted, a cross-sectional and a longitudinal. The cross-sectional, we had 73 participants at around the age of 12 years. When I refer to sex, or female, male here, I refer to biological sex. We do measure gender, but we haven't incorporated that into these studies. The cross-sectional, slightly more males. In the longitudinal study, 136 participants, average age 14, slightly more females in this. The longitudinal data involves 720 data sets, so on average 6 time points in this analysis, but ranging 3 to 14 time points. For each study, individual FA maps are registered with standardised DTI templates, and we create study-specific templates, cross-sectional and then longitudinal, and we use TBSS, track-based spatial statistics, to determine our DTI metrics, FA, RD, AD, and MD. And in the cross-sectional paper, we utilised TBSS to identify significant clusters correlating with social connectedness. Whereas in longitudinal, we've modelled the trajectories of the DTI metrics according to social connectedness group and then disaggregated that according to biological sex. So in terms of findings for the cross-sectional, we identified 13, We identified 13 different tracks that were correlated with social connectedness. Among them, 6 tracks had correlations with all 3 DTI metrics. And the most significant in terms of correlation coefficients was the corpus callosum genus, and I'll come back to that. But importantly, what we found across all these correlations was that there was a consistent pattern of these associations whereby decreased social connectedness was correlated with increased fractional anisotropy and decreased radial and axial diffusivity. And we took that as perhaps indicating that those with lower social connectedness had decreased axonal density or perturbed coherence. And that may reflect that these people with lower social connectedness have less developed neural connections or less myelinated axons and differences in their axonal calibre. As I said before, the corpus callosum revealed the most significant correlation, so we ran some follow-up regression models and found that only social connectedness and not psychological distress or sex or age predicted variation in the DTI metrics. So I'm sure many of you are familiar with the anatomy of the corpus callosum, but just to clarify, when we're looking at these bundles of white matter tracks in DTI, in these studies, we're looking at the genus being the front bundles of white matter tracks connecting hemispheres frontally, and then the body and the splenium, which is at the posterior. So the longitudinal study, what we did was we implemented a functional principal component analysis to determine the social connectedness groups. So this was people who were persistently low or persistently high in their social connectedness. And you can see the numbers there fairly even, slightly more, 73 with low social connectedness and high proportion of female versus 63 with high social connectedness and 48% female. And in this paper, or analysis, we just focused on the corpus callosum and those 3 segments, the genus, body, and splenium. And we implemented generalized additive mixed models, which are very good for nonlinear complex data sets like this. And what we did was we tracked or looked at the DTI metrics over time as a whole group in terms of males and females together, low versus high social connectedness, and then we disaggregated the data according to sex. And that little schematic down the bottom there just shows you the sample in terms of the participants involved, the 136, and how many time points of DTI scans and social connectedness measures they had. This is a busy slide. I'll just basically highlight the key point here, that this is showing, now sorry about the colours, it's slightly changed, but in this figure, low social connectedness group is blue and high social connectedness group is red. And this is everyone together, but you can see across the GNU, splenium and body, all three of the DTI metrics, there were no differences at all in terms of the social connectedness groups across time or over this period for any of the DTI metrics. However, when we disaggregated the groups according to sex, significant differences were discovered in females only, whereby there was increased axial and mean diffusivity in the CC-GNU for those with high social connectedness, and particularly in that early adolescent time, so between 12 and 14 years of age. And similarly, only in females, we found that there was increased axial diffusivity in the corpus callosum splenium for those with high social connectedness. This was a little bit later on in the 16 to 17 years of age, but there were no differences between low and high social connectedness across these metrics for males. So by way of discussion, we're seeing some consistency, I guess, between this cross-sectional and longitudinal analyses. Certainly the longitudinal analysis only focused on the corpus callosum, but we are seeing that there seems to be differences in terms of white matter integrity in that early adolescent period, around 12 to 13 years of age, and the longitudinal analysis revealed sex differences that we did not see in the cross-sectional. So basically across the papers or studies, we're seeing that there's a negative association between social connectedness and corpus callosum GNU axial diffusivity in particular. So the question is whether this is a marker of axonal coherence or a maturational issue. And we do know that in other literature, young people with increased peer victimisation or rejection do have changes in their white matter maturation, particularly in the corpus callosum GNU. The mean diffusivity marker being increased in those with social connectedness, with low social connectedness was, with high social connectedness was an interesting result, and perhaps something I need to look at further, but it could reflect an adaptive process, axonal reorganisation or increased tissue complexity, and there's evidence in adolescent data to indicate that. But the key limitation really of these findings is that we haven't measured white matter volume, so we wouldn't like to do that to better interpret these DTI findings. So in conclusion, how many minutes have I got? 2? It seems that the longitudinal analysis is revealing some more fine-grained details, perhaps a clearer association, certainly in focusing on the corpus callosum and the GNU in particular. We know that that segment is important in terms of transfer of information between the hemispheres, particularly the frontal regions. It's involved in processing complex tasks, verbal and non-verbal, and communication, emotion regulation, and social interaction. So obviously important in social connectedness. So overall, across these studies, we are finding significant relationship between social connectedness and white matter integrity, but particularly in early adolescence. And it seems to be also particularly in females. But we do need to investigate things like pubertal onset to get more information about this. And we do know that sex hormones do differentially impact white matter microstructure. How long have I got?
You've got 1 minute.
1 minute, okay. So I indicated that I was going to talk about some sleep findings as well in the abstract, but I won't go into these, just to draw your attention to these papers done by Dan Jameson. But we've been looking at the relationship between white matter as measured by DTI and sleep in the labs, in the longitudinal adolescent brain study, and finding associations with fractional anisotropy and sleep measures. Cross-sectionally, we looked at the relationship between DTI before COVID and predicting COVID-related worry, and we also found longitudinally that poor sleep reduced myelination rate. So if you're interested in that, look at those papers. And by way, maybe a bookmark, I guess, or a bookend, I should say. We heard earlier on about early life stress, just to flag that a project we have right now by Nikki, a PhD student in our team, is looking at early life stress, just published this meta-analysis, but Nikki is recruiting participants in labs at age 16 and interviewing them about their childhood trauma or maltreatment or early life stress. And we measure all those measures that were mentioned before, the Child Trauma Questionnaire and ACEs. And Nikki has interviewed 40 participants so far at age 16 and above, and what we're going to do is look at that, and 40% of those are endorsing 4 or more ACEs, adverse childhood events. So we're going to look at how they differ compared to their peers that don't experience that many ACEs, early life stress, in terms of their brain development. And another shameless plug, just for those of you who are interested in hearing more about longitudinal adolescent brain study data, I urge you to go and chat with Amanda Boyes, who's just here, and have a look at her work in the Brain Age Modelling Project. And I have nothing more to say except to thank the team and the participants especially. We're still going, we're still looking for funding to keep going. Hopefully the study will go for 10 years, or more. It's been 7 years so far. A lot of people to thank, and if people are interested in talking to me and others in the team about accessing data, please get in touch.
All right, we have time for a couple of questions.
Just for me or
Just for Dan. I miscalculated earlier. I don't think we'll have time for questions for the entire panel, unless you want to stay here during the coffee break. I don't think any of us have any other plans.
Hi.
Hi.
Thank you so much. Super interesting talk. I have a very brief question. I'm sorry if I missed it. How you said that you assess gender and sex. I was just wondering, like, how you did that, like, what tools you used?
It was self-report and confirmation from parent about biological sex, and we did ask about other, and none of our participants have reported other in that question. And we've recently added another questionnaire. Well, not recently, but later on in the study, we implemented a questionnaire about gender identity, and so on. I can't remember the name of it, but if you get in touch, I can give you more information about that particular questionnaire that we use. Okay. But it's just biological, sex self-reported and confirmed by parent.
Yeah. But thank you very much for including the very important aspect. Thank you.
Thank you.
Any other questions? If you have questions for the other speakers on the panel, please feel free to come up or ask through the app. We have 2 more minutes. The coffee will still be there. All right, looks like folks are... Okay, all right. Well, thank you so much for attending our symposium. We look forward to seeing you at the rest of the conference.